variations, right? So these fit variations survive, and that is exactly is known as natural selection, and that is a driving force of evolution as per the uh, theory of evolution by the Darwin. And these days we know it's not just the natural selection but also drift, right? The so-called neutral theory of evolution by Moto Kimura, that's also popular. All these are cladistic method. Now coming to the older phonetic method, we still use that in phylogenetic reconstruction, so-called distance-based method. Like neighbor joining method, right? So, and also this one is really faster comparing to cladistic method, which is very, very slow. So that's why phonetic methods like neighbor joining or UPGMA is a good starting point. And especially if you are working with the whole genomes, then you can do this work for the first time and then go to the cladistic method. So cladistic method, a good example would be maximum likelihood. And as I told you, cladistic method is inferred by looking at its evolutionary relationship. So it's not just the current data but also the past, you know so yeah, that is what the it construct the resulting tree is known as cladogram, while in the earlier one phonetic method, it is phenogram. So there is a difference between that. A phylogram and cladogram is quite same. The only difference is that cladogram, the branch lengths doesn't make any sense. Only the branching order makes sense. But in phylogram, even the length of the branches makes sense. Well, we will see all these terms in depth. Don't worry about it. Okay. So now you can see that these two are a representation of two trees. Here you can see that is angular represented. So cladograms are usually drawn like this, angular, where only branching order makes sense. So from this we know it all bacteria are actually doesn't evolve from one common ancestor. You see, it's not a cluster, right? But all eukaryotes did evolve from one common ancestor, isn't it? So yeah, well, bacteria 2 and 3 evolved from one common ancestor. That is also fine. Group like this, the bacteria, this is known as paraphyletic group, because you need to add something else to make it a monophyletic, right? So anyway, in this case, the branch length doesn't make sense, only the order makes sense. But in this case, the bottom image, which is something called phylogram, even the length makes sense. The length is proportional to the substitutions per unit time. Basically, it is proportional to the evolutionary distance. So, for example, eukaryote 2 is so long, isn't it, compared with eukaryote 3. So, we can say that eukaryote 2 accumulated more evolutionary changes, that is, more mutations than eukaryote 3 from its common ancestor. So those kind of references you can draw from looking at these two trees. So the discipline of phylogenetics is it's a study of evolutionary relationship between the organisms. So yeah, it's basically evolution. Study of evolution these days is known as phylogenetics. And an important aspect of phylogenetics is systematics. The taxonomy and systematics. The so-called phylogenetic systematics is the gold standard on botanical classification these days. So uh, any botanist should know phylogenetic systematics if you want to claim that you are a botanist, isn't it? Well, the field was developed by Willy Hennig. He was a German entomologist, the one studying about insects, isn't it? In 1950, he coined this term as well as the discipline of phylogenetics. So, well, phylogeny, as you know, it's a tree-like representation of genealogy or the evolutionary history. Here is an example of the whole biodiversity of the earth, the so-called tree of life. So the zoomed out tree of life is something like this. It has three domains: bacteria, archaea, and eukaryotes. As you can see that archaea and eukaryotes are monophyletic. But if you define bacteria and archaea as a prokaryote, so well, prokaryote does not form a clade or a monophyletic group. I hope it's clear. Well, eukaryote is monophyletic, right? So this one is by the Carl Woese. So Carl Woese was an archaeobacteriologist from the US. So after this tree became very popular, now we have yet another latest system, which is Six Kingdom system of Cavalier Smith from Oxford in the UK. That is also a tree of life. That is a bit more modern approach. Okay, so the tree of life, as you know, this is a very famous image of the Darwin's own notebook. I think this is how the evolutionary tree looks like, is his own handwriting. So this tree is exactly what the phylogenetic tree is. But remember, during the Darwin's time, no one knew how to make trees like that. But he still imagined a tree of whole living beings like this. You see? Well, talking about evolution, there are divergent, parallel, and convergent evolution. I hope some of you are aware of this thing. Almost all students are aware of it. While divergent evolution is the real evolution. You see, it leads to homologous structures. Homologous structures are, or traits are, morphologically and sometimes functionally similar because of the shared ancestry. At the same time, homologous structures can be plesiomorphic or apomorphic. This is very important. Plesiomorphic means present in the common ancestor and all its, some of its descendant, not all the descendants, right? Apomorphy is the every single descendant, it should have that character, right? While plesiomorphy is some have, some did not have. So these plesiomorphic characters are ancestral character, while apomorphic characters are derived characters. So modern systematics use only apomorphic character, not the plesiomorphic ancestral characters. Right? Here is an example. Poikilothermy, that is basically cold-bloodedness, or ectothermy for the common ancestor of reptiles. You know, well, common ancestor have, there is an ancestral character, and some of the descendant have, like reptiles have, but some other descendant do not have. For example, birds don't have it. Right? Birds are not cold-blooded; they are warm-blooded. Right? While synapomorphic is every single of the clade, every single of the descendants have it. For example, a mammal is a synapomorph. Mammal is a monophyletic group defined by presence of mammary gland. Every single descendant have it. At the same time, convergent evolution leads to analogous structure, look-alike structure. You see, while there is no common ancestry, but the structure look quite similar. That is exactly what a homoplasy means. Okay, same form. Phylogeny attempt to reconstruct the pattern of divergent evolution only. That is very, very important. Phylogeny, there is an assumption going on in phylogenetic reconstruction that evolution is due to divergent evolution alone, not due to convergent evolution or parallel evolution, right? So that such things are invalidated in phylogeny. So only the divergent evolutions are considered. Okay, so that is why if you choose certain character which are convergent to construct the tree, then the trees are invalid, right? So it is extension of the common descent postulation of Charles Darwin, right? The phylogenetic trees. And you might know that these are all hypotheses, right? So yeah, there is no one can know exactly what really happened. So it's something like looking at the post-mortem, doing a post-mortem, and understanding how the evolution happened in the past, right? So true evolution is a divergent evolution, isn't it? While convergent evolution is pseudo evolution, it doesn't happen in the nature, but we think that these two structures are related, but it is unrelated lineages, like wings, for instance, wings of insects and birds, which have no, it is not related in evolutionary point of view, right? So these structures are analogous or homoplastic, right, or homoplastic, that leads to polyphyly. For example, here you can see that presence of six leg. While if you make a group of animals which have leg and animal which do not have leg, like snake, you know, well, that is a homoplastic. It is not a natural group at all, right? Even Insects lug is not really lug, it's just an appendage, isn't it? So here is another thing. Winged animals, animals with leg and no limb. So this character is also paraphyletic group. As you can see that this is a monophyly here. Insects, sauropsid and mammal, right? So yeah, polyphyletic group is basically what you call homoplasy. And polyphyly means many ancestors, right? Now, apomorphy and plesiomorphy, I introduced this term, very, very important term. Apomorphy is the very important for the phylogeny. In fact, only apomorphy matters in phylogeny, right? Or DNA taxonomy. These are specialized or derived or changed character state or innovations, evolution innovations, okay, apomorphy. And when this character evolves through multiple lineages, the descendants of the common ancestor is called synapomorphy. Every single has this character. Okay. Yeah. Just a minute, please. Okay. I'll be joining right back. Okay. Please follow. All right. So I hope I'm audible. Can someone please confirm?
Yes, sir.
All right. Yeah. So, apomorphy is basically an innovation in the phylogenetic tree, and the common ancestor and all its descendants have it. For example, mammary gland is an innovation, a derived feature, a new feature, and every single mammal have it, right? So, and every single descendant, when this is being shared, that is called synapomorphic character. So those traits, a new species has to be defined based on apomorphic character or synapomorphic character. I hope it's clear to you. So it has to be something new, not existing in the ancestors. So yeah, for example, pollen grains, if you see, tricarpate pollen grain is a synapomorphic character for all the angiosperm. So every single angiosperm pollen, if you see, the pollen has got three culpa, right? Tricarpate. So the grooves, isn't it? So yeah, that is an evolutionary innovation. So that is called apomorphic character, right? So monophyletic groups are those groups defined by this synapomorphic character or synapomorphic character, right? While polyphyletic or paraphyletic are not defined by synapomorphic. These are defined by plesiomorphic character, right? Plesiomorphy leads to paraphyly basically. So plesiomorphy is the ancestral character, right? Original or primitive character is known as plesiomorphy. So if you use ancestral character to define a new one, then the group are something called paraphyletic group. Okay, so that is what monophyly and paraphyly, that difference here. For example, if you look at the the animals, isn't it? If you see that the textbook, old textbook, even today, school textbook, there is something called reptilia is being taught, isn't it? One of the phyla, animal phyla, reptilia. So reptilia is defined as all the descendants of amniota, that means right here, right, except mammalia and apes. So you're defining a group with certain exception. Such groups are known as paraphyletic group. I hope it's clear. Or another way to think is that if you think of this tree as trees made up of some metallic wire, and paraphyletic group, to separate you need more than one cut. So if it's monophyly, the entire group is a monophyletic group, you can cut by just one cut here, isn't it? But for paraphyly, just one cut is not enough. I have to cut here, the first cut, then second cut and third cut, because there are exceptions here. That is called paraphyly. Here is another example which we just discussed. Here is basically the yellow one is called similiformes. New World, Monkey Old World, apes, and human. All these are descendants of one common ancestor, most recent common ancestor, MRCA, right here. So this one is a monophyletic group, absolutely fine. Now look at this group. This is the light blue shaded group. It's called prosimy. So prosimy is traditionally accepted taxonomists, traditional taxonomists of 19th century early on. They thought there is a group called prosimy, this group. While this group is, now we know it is a paraphyletic group. So it is not a natural group at all, right? So why it is a paraphyletic? Because to isolate this group, you need two cut, one cut here and one cut here. But for this, just one cut. Now this one is a polyphyletic group, right? Polyphyly means many ancestors, right? It doesn't have any common ancestor, right? So here is another example. Aves and mammalia together, if you group a group, there is a polyphyletic group. And this we already said reptilia is a paraphyly, while the entire thing, this entire of this one amniote is a monophyly. So latest systematics, right, have names of the group. All these names are only monophyletic. So paraphyletic and polyphyletics are excluded. These are invalid in the latest systematics. Here is another example of a tree. So in this tree, here, this label, this one is a monophyletic. What does that mean? This red means a group encompassing A, B, C, and D with a common ancestor here. Right? This is a monophyletic group. Now, this is a paraphyletic group, a group encompassing taxon C, D, E, and F. Why it is paraphyletic? Because B and A is excluded. It should be part of this entire group, right, to make it a clade, isn't it? Now, polyphyletic is just member randomly chosen from various branches. That is called polyphyly. I hope it's clear. Now, coming to the DNA, it could be homologous or analogous, right? The divergent evolution results in the homologous sequences can be of two types, basically orthologous sequence and paralogous sequence. So orthologous sequences are sequences separated by speciation events. The DNA sequence separated by speciation event, like for example, Homo sapiens and Homo neanderthalensis, right? Same gene COX1. So the COX1 of Neanderthal and COX1 of human being is called orthologous sequences, right? While paralogous sequences are sequences or DNA sequences separated by gene duplication event, like hemoglobin of mammals and leg hemoglobins of leguminous plants. So these are a classic example of gene duplication, right? Or uh, uh, globin A and B subunits. All these are examples of the paralogous sequences. So paralogy means this one is because of the duplication of the genes. Right? Genes get duplicated, and the gene duplication is a very important step on the synthesis of new or evolution of the new gene, right? The old gene copy becomes a proto gene, right, which become redundant. The new one evolve into to have new functionalities, isn't it? That is how the genes evolve. So ortholog versus paralog, the difference here is that orthologs are separated by speciation, while paralogs are separated by gene duplication. So if a phylogeny includes both of ortholog and paralog, it is absolutely incorrect. Okay. So for example, here, this one, the globin gene, early globin gene, there is the first event here is, the gene duplication, alpha chain and beta chain of the globin. So among the alpha or beta chain, rat, chimp, and human. So these are orthologs. These are sequences separated by speciation events, right? So yeah, so you should not include both together in the same phylogeny. Yeah. So yeah, that is how. Now coming to gene tree versus species tree, what is the difference? So gene tree is basically what you construct in the phylogeny. right? Trees based on the evolutionary history of one gene or multiple gene is known as gene tree. While species tree is the real tree. Like if you believe in God, only God know what really happened in past, isn't it? So all the trees that we make are nothing but approximation. These are all hypotheses, right? Nothing is certain. So the only certain way to come up with species tree is by doing a computational simulation. So we are starting from non-tree and we are actually doing back in time. So that kind of simulation is the only option for it. So lineage sorting, this term means incongruence between species tree and gene tree. So you can see that there are two genes here. Gene, this blue gene and red gene. So the evolutionary history of both of these are not same. That is what, right? So in the blue gene, you can see A, B are more similar than C. But in red gene, it is B and C are more similar than A. So these two genes, if you see the separation time, you can see that the incongruence between these two, uh, trees, that is exactly called the lineage sorting. But if you see the true phylogeny and the other two genes, the green gene and red gene, so well, what green genes speak is a different evolutionary history. The story is different, right? One and two are similar, while red genes say it is one and three are similar. But the true one is like, it is neither of these two. It is basically two and three are similar than one. You see, so these are completely incongruent. The separation event where it is happening and how these are clustered together. So that is all different. So that is what you call as lineage sorting, right? So, and also in the case of lineage, well, lineage sorting could be because of the paralogy, that is gene duplication event could have happened before the speciation event happened. Here you can see the alpha and beta chain separated before, then speciation events of fugu, mouse, and human separated, isn't it? Now coming to something called molecular clock hypothesis. So basically, the molecules like DNA and protein, that is amino acids, if you look past in time, so the mutations happening at a constant rate in especially in DNA molecule. That is what the molecular clock hypothesis is. So if you see that today the two plant species, how many mutations are separating between these two plant species? For example, if you see one gene, let us say RBCL gene of two plant species, how many mutations are there, right, between these two plant species? So if you know this one, you can actually make a modest estimation when exactly these two species split in the tree of life. So yeah, like how many millions of years back, right? So in that way you can construct a phylogenetic tree with time axis. So exactly the splitting happened how many millions of years back. So that is application of this molecular clock hypothesis. you know well, molecular clock means, well, relaxed molecular clock is a bit more stringent. Here each branch can have its own the rate of evolution. So rate of evolution is not constant across the tree. uh, the branch can have its own evolutionary legacy too. Okay, so to summarize, the evolution can be divergent and convergent. These are the type of evolution. While coming to the trait, it could be homologous or analogous evolution, right? So the homologous traits are resultant of the divergent evolution, while analogous traits are because of the convergent. Now, homologous in the character state could be synapomorphic or simpliciomorphic. Synapomorphy lead to monophily, while synapomorphy is evolutionary innovations or derived character, shared derived character that leads to monophily, while plesiomorphy is ancestral character, shared ancestral character, simpliciomorphy leads to paraphily. While talking about sequences, DNA sequence could be orthologous or paralogous. The difference is the orthologous are sequences separated by speciation events, while paralogous are sequences separated by gene duplication events. Right? On this branch, homoplasy leads to polyphyletic clades. Okay, homoplasy is the character state because of the analogous trait or divergent evolution. Okay, so well, I wrote this article in Resonance of Indian Academy of Science, a two-part series on tutorial on phylogenetic inference. You can just check out this one. It's just a thorough overview of how all these concepts, right? In case you want a bit more in-depth coverage. Now let's start with our next important topic, plant DNA taxonomy. So first we'll see the difference between molecular systematics and DNA taxonomy. So well, it's pretty similar, right? Molecular systematics is systematics based on molecules. So it need not be only DNA, but also amino acids. While DNA taxonomy is a subset of molecular systematics, right? We are looking at only the DNA sequence. So taxonomy based on DNA sequence is called DNA taxonomy, isn't it? So DNA systematics, as I told you, is a better term. Phylogenetic systematics is synonymous to. Okay. So all these methods uses barcode, right? DNA barcode or molecular locus or molecular marker. All these terms again are synonym. Basically, this is a short stretch of the DNA sequence that is employed for the molecular systematics or DNA taxonomy. So what we are amplifying and comparing with the database to construct the tree. So these are known as locus or barcodes. Okay. Now these two terms, barcoding versus DNA taxonomy, what is the difference here? DNA barcoding, the term is only to identify the non-species. If the species is already known, you can identify by DNA barcoding method. Barcoding is especially used for authenticity testing, like in Ayurvedic medicine, claiming that it has Brahmi plant. Well, how sure you are that it has really Brahmi? Isn't it cheating? Some people, some companies can adulterate, right, with inferior plant substitutes, isn't it? So you can do a DNA barcode on it to make sure that it is Brahmi, really. Or if you are doing a field survey and you're clueless what plant is that, so you can do a DNA barcoding, especially when the species is very plastic morphologically. Plastic in the sense that morphology keep on changing with respect to various ecological pressures. For example, in marine plants, the algae which I work on, some of the seaweed growing on on sheltered habits, that means habits without much of the waves, have a different morphology than exposed rocky shores. Though those are exactly same species, but morphologically completely different. One could be branched terete structure, while the other one is having frond-like appearance. So the only option you have to see is it really same or different is to do a barcoding approach. Now coming to DNA taxonomy is not just to identify. Of course, you can identify, but beyond that, you can do some significant taxonomic revisions like discovery of a new species, species discovery, or new species description of new higher taxonomic higher taxa like genus or order division. Why not? All those things are part of the DNA taxonomy. Okay. So using DNA sequence for taxonomic or biodiversity characterization for species discovery and delineation is known as DNA taxonomy. So basically using DNA for taxonomy. So you contrast that with DNA barcoding, which is identification of the previously described species by public databases. So DNA barcoding is something like barcode that we use, like you go to a supermarket, right? A big supermarket, I'm sure all these have got barcodes of every product. So they have a laser scanner, and the reason why they have laser scanner is to speed up this checkout process, isn't it? So same way in taxonomy also, the barcodes speed up the identification, and the same analogy with the supermarket. These barcodes, scanner on the barcode will work only if the item is entered in their tally. Software tally is used usually in every supermarket. So if that product is not there in the tally, then no, it cannot be used, isn't it? So the same way DNA barcoding works only if there is already some barcodes existing in the database. Otherwise, it is of no use, right? So both relies on sequence data, standard barcodes, and generated barcode data will be searched against the database, right? So the database is basically, usually we all use NCBI nucleotide database. Okay. Well, many countries have their own database, like DNA database of Japan and all, but all these are linked together. So there is no point in searching specifically in Japanese database, right? If you search in NCBI nucleotide, you will be also getting the accessions of the DNA database of Japan, right? So that is why no point in searching in every single database of the world. So the way it works is that first you search in this database, like the NCBI nucleotide, to see, or something called blasting, right? Your sequence, you're blasting with, you're performing a blast with the NCBI, nucleotide, and then is there any match or not? If there is a match, then you already describe, okay, this is a non-species. For example, you isolated a random plant, then you did DNA sequencing of it, and then you matched with the database, and it turns out to be mentha, the mint. So 100% match. So then you already identified it. So it is what you call it as species identification, isn't it? Now you do an expedition in, let us say you're going to Araku Valley, right? And then you collected some nice orchid in Araku Valley, and you have no idea what it is, right? You extracted the DNA, you did an RBCL, you know the gene RBCL, you amplified and sequenced, and then you blast with the NCBI nucleotide, and there is zero match. Then what would be the reason for it? So two reasons could be. The first reason is that species in question is already described by some taxonomies. For example, a very old botanical professor from Andhra University have already described that orchid in Araku Valley, but he or she did not sequence it and submit that sequence in the NCBI. So if that is the case, then you cannot claim it as a new species, right? So, yeah, the second reason could be that species in question is truly new, previously undescribed. So that is what almost everyone or all taxonomists want to prove, isn't it? That the plant is a new species. It's a discovery of the new species. But you cannot automatically conclude that new species discovery. You need to look at the first option. Make sure that it's not the case with the option number one. So to rule out that first possibility, a taxonomist first have to list out every single species of the genus. So whatever the Orchidaceae, inside the Orchidaceae, you have to list out every single species known, and are you sure that every single species have RBCL data available? So if it's not available, these are gaps, gaps in the DNA barcode you need to generate. And make sure that your new species, the purported new species, do not match with any of the non-species of that genus. That is very, very important step, right? And even after that work, still you didn't get any significant match with all non-members of that genus, then it's truly novel. You can, by all means, you can claim that it is a novel species, and you can write the paper and get it published in a good journal. Okay. So what constitutes significant match? That is also very, very important. So if the match is, like, let us say 98% with a non-species, can you call it as a new species? So, well, there is no consensus exist, right? With what you call it as statistically significant match. But nowadays, the consensus seems to be emerging among the, eukaryotes. Like most of the plants that botanists work with are all eukaryotes, isn't it? Except blue-green algae. Yeah. which is a prokaryote, cyanobacteria. So for eukaryote, it is 98.7% is the currently accepted cutoff. So if you're getting a match less than 98.7, let us say you're getting 98% similar to a non species in the database, then still there is a chance that it might be truly new. But in case you're getting anything above 98.6, that is 98.7 or 98.8, then you cannot claim that it is a new species. It's already a non-species. While the cutoff is much more lesser in the case of prokaryote, it is 95%. So it is really difficult to discover a new bacteria because of the stringent cutoff. 95% is the cutoff, not like 98.7. At the same time, bacteria, the species diversity is a lot more than eukaryote. So in reality, it is comparatively so much easier to discover a new species of bacteria compared with the eukaryote. Okay. Yeah. Well, the species concept, I'm sure you know different kind of species concept like ecological species concept or biological species concept. The latest one is called phylogenetic species concept. So based on monophyly. So whenever a group is monophyletic, remember that the monophyly. Monophyly is the groups defined by synapomorphic character or a group with one common ancestor and all its descendant without any exception. That is called monophyly. So if in a tree, if you see a monophyly, that monophyly is if the all descendants of the monophyletic clades are the same species, then that species is a new species as per phylogenetic species concept. Right? Now, how do you choose the barcode? Which barcode to amplify? Isn't it? So that is where this approach comes in play. Tortoise and hare approach. So tortoise, as you see, it is so slow, right? Slow in walking, while hare is super fast. So likewise, certain genes are very slowly evolving, especially genes. No? Any kind of genes, the evolution rate is slow. Why not fast? Because fast mutations in the gene will more or less likely invalidate the descendant. That is, baby will not be born, or offsprings are born with certain genetic abnormalities that render them unfit for reproduction and passing that gene to next generation. So that is why these genes are coding DNA sequences. Basically, it has the phenotypic characters, isn't it? So yeah, so if something abnormal happens to a gene, it will have huge ramifications on reproductive success of its offspring. So that is why genes are so much slowly evolving compared with fast evolving loci like intron, right? Intergenic spacer or intron. Inside one gene, there will be introns and exons, isn't it, in eukaryotes? So introns are locations where mutations are tolerated because introns are spliced off before getting translated, right? After transcription, post-transcriptional slicing happens. So that is why these introns do not code for any functional proteins. So that is why the rate of evolution is so much higher in intron. So you can use introns like ITS for lower taxonomic level, while slow evolving loci like genes are good for higher taxonomic level. What does that mean? Lower and higher taxonomic level. Higher means like domain, kingdom, right? Phyla, class, division, all those are higher level. While lower taxonomic level is like genus, species, or subspecies or variety, right? Even population geneticists are looking at the haplotypes, right? Within one species, how different gene variants are shaping. All those things, to do that, you can rely on intronic sequences like ITS sequence, okay? So that choice depends upon your goal, what kind of, at what level of the taxonomy you're working with. So these are some of the most important loci used in DNA taxonomy. Basically, these are barcodes. For plant DNA, you see that these are all the barcodes which we use, like ITS. 18S for all eukaryotes, which is a nuclear. 16S is used for blue-green algae, the cyanobacteria, and other prokaryotes like bacteria. RBCL is used. TRNLF intergenic spacer is used for plants. So both of these are in the chromosome, I mean, in the genome of the chloroplast. And the first two are from the nuclear genome. These are chloroplast genome. Now, cytochrome B and CO2ND1, that is cytochrome oxidase subunit 2, NADH dehydrogenase subunit 1 is a very commonly used barcode for animals, which is there in the maxi circle of the mitochondrial DNA. Cytochrome B is also in the mitochondrial DNA. MATK, maturase K, is used in plants, which is encoded in chloroplast genome. So yeah, there are so many barcodes available. So another very important criterion is that if you're working at certain level, which barcode is already available? That is a very, very important question, right? So in case there is no barcodes available, let us say that you're working with orchids of Araku Valley. So there are no barcodes of RBCL exist in the database, while there are a lot of 18S existing. So despite that, you're going ahead with RBCL. You sequenced it, you generated a sequence, but then virtually that sequence is of no use because there is nothing against which you can match with. So that is why it makes more sense to look at NCBI taxonomy and NCBI nucleotide to see which locus is already barcoded and most barcoded, right? Yeah, so for example, Brahmi plant, if 18S has maximum number of accessions, then you should stick with 18S rather than going with the MATK or TRN, LF spacer, all those things. There's no point in going for it, right? For example, if you're working with red seaweed, red seaweed like Kappa, Carrageenan, right? Carrageenophytes, right? Yes, so, these like Hypnea, for example, you're working with Hypnea, and what barcode can you do it? Of course, you can amplify either RBCL or 18S or ATS. So many things are TRNLF. But if you search in the online database of Hypnea or any other red seaweed, you will know that all this red seaweed, the predominant marker is cytochrome B. So yeah, COX basically, right? So although it is used for the animals predominantly, but among the red algae, like Porphyra. So COX is the most often used locus. So it makes more sense to stick with the same locus than going with something entirely new. Right? So these are some of the most common markers. As I told you, this term marker is the same thing as locus or barcode. Could be from plastid or mitochondrial or nuclear. So for green algae, tough and RBCL is commonly used. Red algae, it is COX1 is the most commonly used. Also COX2-3 spacer, right? Also RBCL, Rubisco spacer are also being used, but this one is the most commonly used. Brown algae also COX1 is the most commonly used, while others are PSP-A, RBCL, Rubisco spacer, and RDNA ITS and all. For these are not that very common. For diatom, it is basically RBCL and COX1, both are used quite often. Dinoflagellate, it is PSB-A, and 23 as RDNA COX1 COB. So for the mitochondria, right? Haptophyte, rhapidophyte, these are not that very common. For this one, the euglenophite, it is small subunit ribosomal DNA and large subunit ribosomal DNA is used. Okay, so these days we do something called multi-local phylogeny. So instead of relying on just one locus, we choose one from plastid, one from mitochondria, and one from nuclear for any group that we are working on. For example, if you're working on brown algae like sargassum from India, so it makes more sense not to rely only on COX1. So you choose COX1, also choose RBCL, you also choose ITS, so that you are getting the barcode from 3 different genomes. So why are you doing that? These are all to increase the precision of your hypothesis. As I told you, phylogenetic trees are nothing but hypotheses, isn't it? So to increase the precision, you can do resampling. So all these are resampling methods. So as I told you already, so selection of loci depends on many factors. So one factor is that PCR, which primer works for the PCR and which actually do the sequencing, right? And barcode coverage in GenBank is another very important, which I already explained to you. Which barcode of that species is existing in the GenBank, and also maximum accession number. So stick with the same barcode, right? So ideally, there should be hundreds of other accessions to which your query sequence should compare against. Otherwise, it's going to be tricky. So if you decide to amplify a gene, like coding for superoxide dismutase, so a unique gene you're simply doing, and well, there is no sequence available. There is no point in that. It's virtually useless as a DNA barcode. So don't do that. Okay. So, well, analogies like the DNA fingerprinting in the forensics, right? So, yeah, if you do, if you suppose that you get a blood sample at the scene, a murder or whatever, the homicide case, and then the blood you're extracting the DNA and amplify, the certain barcode, for example, let us say microsatellites. Okay, you amplify the microsatellites. Well, now what will you do? So unfortunately here we don't really have, India don't have that kind of a repository or a database of the barcodes of non-criminals, especially criminals on parole. So world over, criminals while serving in jail term, they go on take parole and then go out and then commit the crime again, right? So to find them, you can do this DNA fingerprinting, right? So well, we really don't have that kind of database, unlike Japan or European Union or America have, right? So that is why it's of no use to do that, this kind of approach here. While, well, it still makes sense if the person is could able to remember, and there are some certain suspect in the case. Like, let us say there are 25, the police made 25 suspects, and you want to cross match the blood in the same with all the 25 suspects. You can do the cross matching with the DNA fingerprint without relying on a centralized database. It is a local search, not a global search, right? Yeah. So what is species delimitation? Species delimitation is that, well, let us say that you did a biodiversity survey of orchids in Araku Valley, and you got a lot of species, lot of sequences, lot of DNA sequences. Now the question is that, is it how many species are there in these sequences? Is it 2, 3, 10, 15? So based on the similarity, you're calling how many number of species are there. That is called species delimitation. That is defining species boundaries from the DNA data, right? Yeah. So let's say that you generated hundreds of sequences at a particular locus. In the tree, these sequences form 4 well demarcated clade. Does these clade correspond to 4 species or not? That is a very interesting question, right? Yeah, so let us say giraffe, right? All around the giraffe till maybe around 7 years back, till 7 years back, and even today in the school textbooks say giraffe is just one species, right? So something happened around 6 years back. So these zoologists, rather phylogenetic systematists who are working on zoological classification, they collected the blood from all giraffes and then they constructed a tree. So in the tree, you can see that there are 4 clade of the tree, well demarcated 4 clade. So then they wrote an influential paper which argues that there are 4 distinct species inside the giraffe. It's not one species. There are 4 species in it. So that is why this phylogeny is extremely important. It's very powerful to see this kind of cryptic speciation. Cryptic speciation means secret speciation. Previously thought to be one species, but within that species there are so many species, distinct species, not just varieties, but species available. Okay. Yeah. So if you see how speciation happened, ancestral species evolved to species A and B, mainly because of a geological barrier, isn't it? Gene flow is impeded by some barrier. In the classic example, this example is called allopatric speciation because of the vicariance or geographical barrier. So, like for example, the ancestor of Homo, right? So then now we separated this one around 3 million years back to Homo neanderthalensis and Homo sapiens, right? So just after the separation, the earliest species concept which you can actually detect is called genotypic cluster concept. Some clusters are being formed. Then comes morphological species concept. So by this time, so this is the time, so after some millions of, after thousands of years, it all depends on which group you're looking at it. Then morphologic distinction comes up, like cranial width and cranial length. If you're looking at the neanderthalensis and homo sapiens, or jaw, right, all these are the reliable character. So after morphologically distinct features come up, ecological species concept can be applied. Ecological species concept means ecological adaptation. Neanderthalensis are more carnivorous, right? The European Neanderthals are mostly meat eating, while Homo sapiens were omnivorous, not just the herbivory, but also occasional animal, but mostly plants, isn't it? So ecological species concept can be used. Then come biological species concept. So you see that till then, Neanderthal and Homo sapiens can able to interpret, you know so well, that is what the latest genomic evidences suggests. For millions of years we kept on mating with each other, neanderthalensis and homo sapiens. But till this moment, so at this moment, biological species concept can be applied, which is completely reproductively isolated. And after many thousands of years, you will be getting monophyletic DNA. So if you are seeing this monophyletic DNA, this is the gold standard evidence for defining a species, not just the reproductively isolated. Earlier, we thought reproductive isolation as the best option to define a species, so-called biological species concept. So now we know it is not that much accurate. The best option is monophyletic DNA in phylogenetic species concept. Well, statistically speaking, there are so many species delimitation methods are available. Depends on, is it single locus or multi-locus, like ABGD, automatic barcode gap detection, spider, statistical parsimony, GMYC model, you know, and species delimitation, BPP is a Bayesian method and all, or Merera method. So, well, these methods, well, so basically you just need to choose any of these method. Well, certain methods are advantageous, while certain methods are only applicable in certain situations. So these methods can detect the delimitation, the boundaries between the species. So environmental DNA, like you go on an oceanic cruise and you collect the plankton and then crush all the DNA and then do a metagenomic sequencing, the environmental DNA sequencing. And in that DNA, well, we have so many of the DNA sequences, right? How many species are there? Is it 1 million, 2 million? No. So you can do this kind of approach here, this statistical tool, right? Well, yeah, so ABGD, there is a web page itself. You can simply go and check out. You can just paste your sequencing faster and then click go, and it can automatically detect the gaps. So what is barcoding gap? Gap is the observable frequency distribution of intraspecific and interspecific distances. So the idea is that genetic distance, interspecific distance means distance between two species, that should be high. Intraspecific distance means distance, genetic distance between the members of the same species, it must be low, right? In order to call it as distinct species. That means, like, let us say human beings, right? All human being inside the Homo sapiens, the genetic diversity is very, very low. But human being and chimpanzee, if you see, within chimpanzee also genetic diversity is very low. But if you see human versus chimpanzee, the difference is very large. And therefore, these two are different species. So that is how barcode gap analysis work. Okay, yeah. So in summary, the taxonomy, DNA taxonomy employs the DNA data, while especially the DNA sequence data. That is what the DNA taxonomy is all about. Okay. Now, broader term molecular systematics is not only the DNA but also amino acid sequences, right? So yeah, everything is in. So basically, the DNA taxonomy is like a subset of molecular systematics. Okay. Now, sequence from the sequence of a particular gene tree is constructed, while species tree is the ideal tree. Okay, the gene tree is what we construct. Okay, and by far the phylogeny construction, also known as phylogenetic inference, remain the central dogma of molecular systematics. Right. So yeah, it's all about how do you make the trees, how do you understand the trees and infer the trees. So that is exactly the central dogma of uh, botanical taxonomy these days. So that is why understanding trees is really important. Okay, so this one is the earliest non-tree. As I already told you, this is a illustration on Darwin's notebook, right? This is the earliest non-tree. So tree, also known as dendrogram or phenogram, where only branching order makes sense, right? Usually perpendicular. So cladogram is also a branching order makes sense. It's an angular representation, cladogram. In the case of phylogram, it is, branch length is also uh, important. It makes sense. So it is kind of rectangular. Okay. Now chronogram is a time tree where usually the x-axis is the time. Okay. Rooted and unrooted, there is a difference between these two, rooted tree and unrooted tree. So rooted tree will have an outgroup. Outgroup is a completely different species. We deliberately add to see that which is the ancestor and which is the descendant. Okay. Just a minute. Okay, I'm just getting. Hello.
Sir,
Yes. Yes. Okay. Yes. You're saying something, ma'am?
no, sir.
Okay. Yes. So rooted and unrooted trees. Okay. So the first one is rooted tree. So basically, the rooted tree means which one is ancestor, which one is descendant. Everything is clear. While unrooted tree means you cannot make any such conclusions from it. So yeah. So unrooted trees are kind of not of much use, but rooted trees are much more useful because you can say that which one is ancestor, which clade originated, which is the most recent common ancestor, all those things. Okay. Yeah. So you can see that this is a rooted tree where time makes sense, while unrooted tree there is no such time. We have no idea which one is old, which one is new. But all this we know from this tree is that C and D form a clade. That means C and D species are similar, while A and B species are also similar, right? While in this case we know A and B are similar, C and D are similar, but then this is how it actually the evolutionary legacy is all about. Now you can see that this is another example of the rooted and unrooted tree. So you can see that here this is the unrooted tree: mouse, human, fugu, and drosophila. While if you place a root here in the drosophila thing, then it becomes a rooted tree. So where the time also makes sense. Fugu, by the way, is it's basically a puffer fish. Puffer fish. The Japanese puffer fish is called fugu. So this is an example of an unrooted phylogenetic tree of Miocene superfamily. Okay. So rooted and unrooted trees, you can see that outgroup is a very good option to root a tree. To convert an unrooted tree to a rooted tree, you can deliberately, purposefully add a taxa which is not related to the in-group taxa. For example, if you are working at working for angiosperms, you can choose a gymnosperm like pine to purposefully put the root in it, in that tree, right? The gymnosperm and angiosperm, yeah, the separation is where the root happens, because you purposefully put an out group in it, right? Yeah. So as you can see, this is our unrooted tree. Well, from one unrooted tree, so many rooted trees can be made depending on where you put the root. Okay, this is the puffer fish. So well, so to root the tree, well, the best method which I told you is out group method, and also midpoint rooting. The longest branch length, you can put the dot in it. And the third method is gene duplication rooting, where an event of gene duplication happens in the tree. So you have to put the root in at that event. So that is really important. Okay. Yeah. So as I told you, it is paralogy, right? Not orthology. So paralogy should always be first, then comes orthology. So you can see here, this one is an example of deliberately adding Drosophila in our analysis, so that you are going to get this rooted tree where Drosophila is a thing. So this is called out group rooting, while this rooting is called midpoint rooting. Drosophila, this is a, you can see that the branch length is really long. So the midpoint of that branch length, you're putting the group. The third one is gene duplication, right? Whenever this one is, alpha and beta chain duplicated, that is where the routing has to be at. Okay, just a minute, please. Hello. All right. Yeah. I'm having a talk. Yes. Yeah. So the tree, you see, the any kind of phylogenetic tree can be summarized in text like representation like this one, right? This is called Nevic format, right? So Nevic format use the nested parenthesis to explain the tree. Here you can see BC, there is one bracket parenthesis, then comma, DE, another bracket. So the way you expand is that you can actually choose from any of the interior parentheses. So BC forms a clade, then that clade DE also forms a clade, then this BC and DE are joined, then finally A is joined, right? So that is how you can convert a tree from Nevic representation. Okay. So you can see that bifurcating node is that one node where two taxa is being formed from that node. Basically, these are species, right? Two species are descending from one node is called bifurcating. While this is called multifurcating node, from one node, multiple species is coming, known as polytomy. So polytomies indicate, you don't have sufficient resolution power at that locus. So usually whenever you get this kind of polytomy, that means that your locus selection is wrong. You should have selected some fast evolving locus. You might have choose a very conservative locus, like for example DNA polymerase 3. So if you choose polymerase 3, all plants will be just same, right? There's not much a difference. You're going to get a giant polytomy, right? So that means that your locus selection is very bad, right? Other things in the tree include leaf. The terminal branch is known as leaf, and edge, node. Internal nodes are basically speciation events, and then the root of the tree. You see, so topology is a branching pattern of the tree. Topology, branching pattern, how the trees are being branched. Root is a common ancestor, also known as MRCA, most recent common ancestor. Internal branch, leaf, also known as OTU, operational, this one, no, the leaf, the final one is called OTU, operational taxonomic units are extant, right? Not extinct. The other one is extinct. Hypothetical taxonomic unit, like the internal nodes are extinct. Only these are extant. Extant means currently available. They're living somewhere in the world, isn't it? So node is basically the point of divergence of the two operational taxonomic unit is called node. So every node in a tree is a speciation event, right? So before this event happened, this separation happened, C and D were the same species. Then suddenly something happened and C and D diverged. That is what the node is, right? So a clade is a group of species descended from a common ancestor, clade or monophyletic clade. So clades are super important for molecular systematics and DNA taxonomy, right? Only clades are named, right? Paraphyletic group is like this. You need to exclude something to define that group. That is called paraphyletic group. Like reptilia is a paraphyletic group. You need to exclude aves and mammalia to define that group, right? So polyphyletic group is group from different branches. I've already explained to you this term: monophyly, paraphyly, and polyphyly, right? Now interpretation, all these terms, let us actually see it, right? So if you see that all these terms you already seen, right? The tips of the leaves are operational taxonomic unit, while node and root. So in the cladogram, the branch length also makes sense. You can see that some branches are really long. These are all Drosophila, while some branches are relatively shorter. These are pretty short branch, right? So this length of this branch is drawn proportional to substitutions per site. This is the unit. You see, substitutions per site, right? And phylograms are always drawn in the rectangular fashion. Now, what is this numbers here? 84 percentage, 100 percentage. So these are bootstrap proportions. In the maximum likelihood approach, these are bootstrap proportions. Basically, this says the probability of finding the same group when you repeat that test, right? 80 percentage of time, you are going to get this clade. So that means that statistical support for this clade is pretty good, while statistical support for this clade is excellent because this clade has 100 percentage support. you know so that is what, for this clade support is kind of less, 70 percentage only. For this clade only 59 percentage. So yeah, that is what statistical significance of the tree is by bootstrap proportions or posterior probability. Okay, if you're using Bayesian inference approach, it's called posterior probability. So these are called sister group. The descendants of the same node, internal node, taxon A and B are sister groups. So taxon A and B is sister to taxon C. If you look at this node, right? So monophyly, I told you just one cut, you can separate, that is monophyly, while non-monophyly, in this case it is paraphyly, you need two cuts, right? Mammals and birds are excluded, then you're defining them as reptile is not natural, right? It's not a clade. So yeah, here is an example. Aves is a monophyletic group, while Dinosauria, excluding crocodiles and aves, you know, if you say dinosaurs without crocodiles and birds is paraphyletic, but Dinosauria is monophyletic, which includes or non-avian dinosaurs plus birds, right? Yeah. So now you can see that, well, this is just a cartoon here. The dinosaur 65 million years ago, dinosaurs. Now the chicken as a dinosaur, yes, as per the latest system of taxonomy, birds and dinosaurs together form one group called Dinosauria. So all non-birds, including the chicken, belongs to Dinosauria, because that is a monophyletic group or clade. That's very, very important. Okay, now if you see that, uh, well, I think it is not that much visible to you. You might be, many of the participants must be using, the mobile phone. So this is like a quiz. So, yeah, so I'll just skip those slides. I'll come back in a short while. Reciprocal monophyly is the gold standard to describe a new species, that is species discoveries. So what does that mean, reciprocal monophyly? So as you can see that, here, all these species, right, this blue species are forming one clade. and none of the blue species are part of the clade of red, and the vice versa too. None of the red are part of the blue. And now these are what? The green one. So none of the green is part of any other clade. So as this pink and orange. So this is a reciprocal monophyly, a complete separation of various species. So all these colors represent unique species. Okay, so that is the meaning of it. Okay, so polyphyly is, as you can see, this is basically ulva and enteromorpha among the seaweed, right? The two kinds of seaweed. Usually ulva is frond like seaweed, while enteromorpha is more hollow tubes, right? Earlier we used to call enteromorpha and ulva. So this is one of the influential paper, so which did two types of barcode, RBCL on the right and ITS on the left. So as you can see, ideally all ulva should be part of one clade and all enteromorpha should be part of one clade. But what they got for ITS as well as for RBCL is a mix of all these together, randomly mixing all these together. The last few are basically Conmania, Ulvaria, Umbraula. All these are outgroup. So remember, outgroup is used to root the tree. So it need not be one taxa, it can be more than one taxa too. Okay, so these are all outgroup. Now you can see inside in group ulva, then enteromorpha coming, then ulva again, enteromorpha, ulva, enteromorpha. So it's completely chaotic. So this finding led the team to say that ulva and enteromorpha are exactly same. It's not separate. Okay, so that is a classic example of polyphyly. Here is an example of cryptic speciation in giraffe, which I just explained a while ago. So you can see that it consists of six species of giraffe inside one species. Earlier we thought all these species are just one, but it has got six separate species, with its own unique distribution of the species geographic distribution. Yeah, cryptic speciation is basically secret speciation within one species, which we think that it is just one species, but there are multiple secret species which coming to the light by the molecular systematics. Okay, so this is the paper published in Current Biology. Whole genome analysis of giraffe support four distinct species. That is another. Some analysis say six species, newer ones say four species, but yeah, the current consensus is that at least there are four species of giraffe. So species split versus lump. The term split means earlier putative species is splitted into multiple subspecies, and or subspecies elevated the rank to the species level. That is called splitting, right? So one older putative species into multiple species is called split, right? So that is exactly what you call cryptic speciation. Okay, while union of the older species into one species, the lumping, right, that leads to synonymy, like enteromorpha and ulva are synonymous. So if it is synonymous, then the older name of these two should have the priority. All these are as per the principles of the code of nomenclature in botanical systematics, isn't it? So right now the current code of nomenclature is what? Which code is in force? Can anyone tell me? In botanical systematics, which code is the latest code of nomenclature? Anyone can unmute and speak. Is all well? Are you able to hear me?
Yes, sir.
Yes. Well, it is something called International Botanical Congress. Yeah, so the latest code in force is called Madrid code. So just finished last week. Okay, and many changes have been adopted in Madrid code. Before that, it was Shenzhen code. from Chinese city of Shenzhen, and even before that it was Melbourne code because IBM happened in CBN happened in Melbourne of Australia, right? So yeah, this committee keep on having meetings to adopt new rules on the nomenclature. Yeah. So right now it is called Madrid Code of 2024. You know. So the branching order is what a topology is. Okay, the order at which a branching happen. So one of the very important hallmark is that if you rotate that branch, no problem with the topology. Topology will not change. For example, I'm just twisting at the lowest branch, to make it like this. BCDA become ADCB, not just the reverse. No problem. It's still conserved the way branches are formed, like CD form one group, here also CD form a group. Then here you can see BCD form a clade, here also BCD form a clade. So now this clade you rotate, no problem. So you rotating, it doesn't affect on the tree. Okay, so these kind of trees are called, yeah. So basically these all these trees are exactly same. Yeah, there is no difference in the branching order or the topology of any of these trees, right? So these are known as, technically these are called compatible tree. Compatibility means information carried in these trees are exactly identical. Okay, so yeah, rotation or twisting or the way you draw the tree, for example, diagonal up or rectangular right, circular, diagonal down. Again, all these trees are compatible because the information contained all these trees are exactly same. Pruning is a different activity. Pruning is you're cutting, some sections you're removing it. Again, no problem with the pruning. So if you're simply cutting from a master tree, some branches are cut off, like the with our garden plants also, right? We prune it, right? Rose, we prune. We just cut off certain branches. So again, it doesn't have impact on the topology, right? Branch order is not affected, right? So yeah, it is still compatible trees, right? Both these trees are compatible. For example, here this tree has two mainly big clade, A, B, C clade, D, E, F, G clade. Now you are making another tree, this clade, by chopping off what? You're chopping off D, you're chopping off C, and you're chopping off A. Just by chop off and rearranging, you're going to get this tree, right? So again, these two trees convey the same information. There is no conflict in it. So that is why these can be called as compatible trees, right? So two pruned versions of the same larger tree. Here, this is a larger tree. You cut off to get this tree, while this is the same larger tree, but you're cutting in a different locations to get this tree. So all these trees can be called as compatible trees, right? Compatibility is a very important concept. Merging is basically you're simplifying a larger tree, a master tree. You're simplifying by naming certain clade, like clade H, D, E, F, G clade. You're calling it as clade H, and then simplifying that clade, substituting this clade with that H. So that is called merging. Okay. Yeah. So these trees contain information about the relative age of the nodes that are on the same path from the root. So that is what that is very, very important. So if you look at the tree, if you're looking in one branch, you can make a relative age estimation. Which one lived older, which is more recent, which is ancestor, which is descendant. All those things you can make inference only by looking at one branch, not the two branch. Okay. So for example, in this case, A, B, and C, which one is older, which one is younger? So B and C are definitely younger than A because A is a common ancestor, right? Ancestor, both of these are descendant, like two kids from one parent. So definitely A is older, you can say, but B or C, which is younger? You cannot call C as younger because it is more branch length. Because remember, branch lengths are not proportional to the time in this cladogram. right? So it is impossible for you to make this kind of inferences, right? Yeah. So only in the case of uh, time tree you can make it, right? If you make a chronogram, then you can make the estimate, relative estimates. For example, this is a time tree. It's a chronogram in which you can say that B happened around around, let us say, 25 million years, while C, this descendant of the A, lived around 33 million years back. Therefore, D is younger, C is much older, while A is the oldest one. So to know that A is older than either B or C, you don't need a time tree. A time tree is needed to compare between B and C, which one is younger. So in this time tree, it's very clear that B is relatively younger than C. I hope it's clear. Oh, by the way, to calculate this kind of time tree also, you can do Mega, the software which we are going to introduce in a short while. Okay. So dated fossils can be used as calibration checkpoint in the uh, chronogram. So to make this kind of time tree, you need a molecular clock hypothesis, and much better if you already know certain fossils, some hypothetical taxonomic unit. This fossil is at least 55 million years back, which we are very sure because of the carbon footprint, carbon dating, C14 dating. And if it is dated fossils are available, then you can put hard limit on certain nodes of the tree. That is how to make a time calibrated tree. Okay, millions of years before present. Yeah. So consensus trees means that both of these trees are compatible, isn't it? Two resolved trees can be combined to make a consensus tree, like tree 1, tree 2. You can actually combine these two trees to make a strict consensus tree, right? So this strict consensus trees means that all these branches are available in it, but you need to introduce this polytomies. As you can see that this is a multifurcating node. It's not bifurcating, it is a multifurcating. From one node, three descendants are coming up, right? So that is called multifurcating node, right? So this also explains strength. The statistical support for the clade is indicated by bootstrap score or posterior probability, depends upon the method of phylogenetic reconstruction. For maximum likelihood, we are using bootstrap. For neighbor joining UPGM and all, we are still using bootstrap. But if you're using Bayesian inference, you're using posterior probability. So as you know, probability is 0 and 1, between 0 and 1. So it is like 0.69, 0.98 means very good support, while less than 0.5 is poor support. Okay. So yeah, this I will skip um, for the brevity. Just a minute, I'm getting multiple calls. Sorry for it. Yeah. Yeah. So basically, I'll just skip to this slide now. This is a quiz. Okay. MRCA of mushroom and sponge. So mushroom and sponge. Which is the MRCA? Which node? EADCB. Which one is the most recent common ancestor? Anyone please unmute and speak. Just to know that are you really following or not?
C, sir.
C.
Uh,
I said that you see the sponge and mushroom,
D,
the
sir.
ancestor connecting the sponge and mushroom.
D, sir.
Yeah, D, correct. So this is the MRCA, right? Now, if I ask you to pick the odd one out of this, which one will you pick? A means this, B is this, C, D.
Sir, repeat question, sir.
So you need to pick odd one out. Basically, three trees are compatible, but one tree is different. So which one is the different tree?
C, sir.
C. Why?
Sir, I think it's D.
This one.
Yes, sir. D.
Why so?
Sir,
Can you
because
explain?
if you check the first one, D
Yeah.
has a common root and E and C are connected to it, right?
E and C and D. Yes. E and C are connected to the D. So D
Yes.
is forms a clade.
Yes, sir. But
Yeah.
for the other ones, it's different.
Yeah. What is that? D
Uh,
then E C. This one is D E then C.
yes, sir. E is the clade here for B.
Okay. So as per you, it is D is the exception here.
Yes, sir. Not just considering the D clade, considering G and H clade as well.
Okay. So as you can see that DE clade, if you look at the DE clade, right, this forms a clade here. DE clade, then C comes. So here you can see that DE doesn't form a clade, right?
Yes,
It's
yes.
it's with a C. Well, in the case of DE is a forming a clade. C also DE is a forming a clade. So which one? Now it is become much more clear to you to say
Yeah,
that
yes, sir. A is the answer.
yeah, A is the right answer. Now see that D, E do not form a clade. And that is the only difference between all these trees. So basically D, C and B are compatible. All these trees are exactly the same. It's just that it's twisting, you're basically twisting like H A become A H here, no problem. A and H is forming a clade. B H A B, this one, B, A, H, all these are forming one clade here. That is also fine. Now, all the five forms a clade, G, F, G forms a clade, C, D, E, D, E, C also forms a clade. D, E forms a clade, D forms, so both these are exactly same tree, you're just twisting. But this tree is different. I hope it's clear. Right. So now, the odd one out here, how will you pick the odd one out here?
Sir, is it D again?
D. What is your rationale?
Srivalli, sir.
C. You're saying C or D.
D, sir, D.
Okay. And what is the reason for it?
Here, sir, I've noticed that for if we take A as a common point here, then G declared is forming a bond with BC and D, but here C and D are they're not on the same plane. They're different, even for FG as well.
I see. Well, again, now so see the answer is basically A here. A, why the relative way the C is being claded. All the rest B, C, and D are actually same. If you really look at, in close, you will see that, for example, D, E, F, G forms a clade, and then comes C. C, D, E, F, G forms a clade, right? So yeah, that is why A is the right answer here, not the D. If you see that the why it is the case, right? If you observe it carefully.
Yes.
Anyway, it is being recorded. You can get a get an idea about it. Now the next question. Clade with the name Dicaryomycota comprises all the descendants of the last common ancestor of moral and puffball. So moral here, puffball here. Basically, all these are fungus, isn't it? So various fungus thing. So yeah, so the name Dicaryomycota means moral and puffball, common ancestor and all its descendant. So then which one is not part of Dicaryomycota? That is the question. Bread mold, consmut, yeast, bread mold, yeast, portabella, consmut and bread mold, yeast, consmut and puffball. Which one is not part of it? Anyone? I hope it's clear the question, right? Dicaryomycota, you see the it comprises of all descendants of the last common ancestor of moral and puffball. So moral and puffball, its last common ancestor, MRCA of these two, and all its descendant. That is called Dicaryomycota. Then which one is not part of Dicaryomycota among these choices? Anyone, please tell me.
Bread mold.
Correct, yes. Bread mold is
Yeast.
right.
Yeast.
No, no, all these. See, basically, moral and puffball were
Yeah.
yeasts, right? So
Yeah.
this one and puffball, the last common ancestor is basically right. This node, right? That's called MRCA, most recent common ancestor. From this node, every single descendant is part of it, right? And that is why except bread mold. So bread mold is the right answer here. So now, which of the following said do not form a clade? First, yeast and moral. Option B is portabella and puffball. Option C is portabella, puffball, corn smut and bread mold. Option D is portabella, puffball and corn smut. Option E, all the taxa except the bread mold. So which is
Sir,
not
second one.
hmm?
Second one, B1, sir, option.
B.
Football and football. Difference, sir.
No, portabella and puffball do form a clade, no problem. It's a clade, right?
Bread,
Okay.
bread mold.
Option C. E is the answer, sir.
E is also not an
Option C.
answer. All taxa
Yeah,
except
answer.
that mold forms a clade, no problem. It's clade proper,
Yeast.
right? A is also not an answer because yeast and moral forms a clade. It's a clade.
D is the answer, sir.
D, portable, puffball and corn smut.
C is the
That is
answer, sir. Sir, C is the answer.
C, correct. C is the correct answer. Portabella, puffball, corn smut and bread mold. Right? This one is a paraphyletic clade. It is not a monophyletic clade. It's not a clade. It's a paraphyletic group. So C is the correct answer here. Right?
Okay.
So yeah, it's a little bit difficult to read. I'll just skip it. So I'll go for this one now. Which of the following living organisms are ancestors to the human being? Chimpanzee, orangutan, white-handed gibbon, all of the above or none of the above?
I'll
Orangutan.
go. It's chimpanzee.
Chimpanzee is the ancestor to the human being? No, it is none of the above. See, that is one of the common misconceptions about the evolution, right? So we are not evolved from chimpanzee. No, we are also not evolved from any of the monkey. Many people think like that, right? Human being evolved from monkey. Not at all. All we can say is that human being and current day chimpanzee or orangutan or whatever the monkeys are had a common ancestor. So current day chimpanzee and current day humans, Homo sapiens, are equally separated from that common ancestor. So that is what the tree thinking all about, right? I hope it's clear. Yeah, so which is not necessarily be true. A is a descendant of B. A is descendant of B. Now D is descendant of B. D is descendant of B. E lived after B. E lived after B. Then option number D is A lived after C. A lived after C. Then E. C lived for B.
D answer, sir. D.
Correct. B, you cannot make that kind of, these are in different branches, so not different branches like A and D or A and C and all. You can never make any comparison. But within one branch, you can make, which is ancestor, which is descendant. I explained this in one of the previous slides. So this is also very important while interpreting the tree. Okay, and yeah, so now which is true? Now you can see there are so many things here. Now one is the flowering plant, pine, then ferns, moss, caryophyte algae, chlorophyte algae. This is what caryophyte is. Now the question is, caryophyte is more closely related to pines than chlorophyte. That is the first option. Which one is true? Okay. Option B is caryophyte is more closely related to chlorophyte than with the ferns. Now option D is caryophyte is more closely related to mosses than flowering plants. Which is correct? Caryophyte is more closely related to pines than chlorophyte algae. That is the first option. Then second option is caryophyte is closely related to chlorophyte than ferns. And the third option is caryophyte is closer to mosses than flowering plants.
Sir, B1, sir.
Yes.
Sir, A, sir.
A.
Sir, B1.
Who said A? What is your name?
Option B, sir.
Many options are coming, but I want to know who said A. What is your name?
Money, sir. Chowdada money.
Yeah, Chaudhara Mani. Miss Mani, tell me why you said A is the correct answer.
I just guessed, sir.
Why you guessed? Tell me some rationale must be there. That is very absurd statement, right? Caryophyte is more closely related to pine. See, so many, pine is very, very new now. Caryophyte is more related to pine than chlorophyte. Chlorophyte is very near, very old. Caryophyte and chlorophyte are very old. While caryophyte is more related to pines than chlorophyte. How come, Mani, you guessed it?
Because this
Yeah.
caryophyte is land plants, sir.
Yeah. You are correct, actually. What you guessed is absolutely right. So many people make this mistake by looking at the number of steps to go, from caryophyte to pine, for example, step 1, 2, 3. So people count the number of steps to see the genetic distance. While caryophyte to chlorophyte is neighbor, not just one step to chlorophyte. So these two are related than pine.
Land
No,
plants.
that should not be the way you look at the tree. Okay, so in a phylogenetic tree, all it matters is clade or monophyletic groups. So caryophyte and pine form a group. Yes, it's part of this group, right? So this group, all the descendants of this group are more related than some other member. It's something like your great grandmother, all the descendants of her are related, right? Than great grandmother, isn't it? So the first cousin twice removed is not really that very common line of relatives, isn't it? So one common ancestor and all its descendants are really related. So the true statement is only A. Caryophyte is more related to pines or more related to flowering plants or more related to mosses than chlorophyte algae. I hope it's clear, right? Okay, this one. Now we will start with the practical. So before that, we can just take a quick break for a while because we have been talking for quite some time. Fine.
Yes, sir.
Okay, so perhaps we can uh, break for 15 minutes and come back. Okay.
Okay, sir.
All
Okay, sir.
Thank you, sir, for your engaging and enriching session. Now the platform is open for questions and further discussion.
yeah, yeah. If you have any questions, let's come back here, or we can break and come take few questions. Why not?
Okay.
Yeah.
Okay. Time for a short break. We will reconvene in 10 minutes. Short
We will
break.
join back at 12:30 చెప్పండి. We will join back at 12.
We will join back at 12:30.
Welcome back to most awaiting session, hands-on training session. I request our renowned resource person, Professor Felix Bat sir, to take up the session.
Okay, hello, welcome back. I hope you had a nice short break. Okay, so now we will go to the practical aspect of this today's program, right? So we have seen the theory. Let me share the screen once again. Okay. So the theoretical background with this background, let us actually now jump to the practicals. Okay, so for which we need to open the Mecca. So basically, I will just explain to you how we are going to do. So the workflow in any kind of DNA taxonomy is that basically we first, of course, we have to go for sampling field, right? Collect the plant material, and then we have to do DNA extraction. So for that, it's better to prioritize the young leaf. Young leaves are better than old or stem or any other parts of the plant for angiosperm if you're working with. The reason is that these young leaves are actively dividing cells, and the DNA is so much easier to extract. So yeah, you don't really need really tough agents to break the cell walls and all. So as it is comparatively easier to extract the DNA, and you don't really need to put a lot of stress on the extraction process. This stress like this vortexing, for example. Many of this DNA extraction protocol will ask you to do vortexing. It's better to avoid vortexing because the DNA gets sheared, and if it becomes small pieces, then it will not be amplifiable. You need to have a template DNA, right? High quality template DNA with a very high molecular weight. So once you extract it, these days everybody use the DNA extraction kits by the companies like High Media and all. It comes pretty cheap, and yeah, so kits will definitely speed up the extraction. And well, before everything, yeah, I mean, after extraction, you need to decide which primer to choose. I told you, right? Remember last time. It depends on the query coverage. It depends on the PCR success. It also depends on which taxonomic hierarchical level you are working on. So if you're working at division or order, for example, class and or higher taxonomic level, it's better to go with a slow evolving locus like a gene. While for species or genus level, it makes more sense to go with the intron like TRNH, TRNF intergenic spacer, or ITS intron, right? Intergenic spacer is also non-coding regions, right? So once you amplify the PCR, well, you need to, what is it? You need to purify the PCR products, right? Yeah. So after purification, you need to do a sequencing, DNA sequencing, right? So post PCR, the cleanup, we usually use centricept spin column. That is one option for cleanup. Yeah, so that is one of the standard option which is available. Or exosap IT is also used, right? Exosap IT is basically shrimp alkaline phosphatase. So you can do a sequencing reaction. Basically, it's chain termination, right? Big dye chain termination reaction, and then you can do a electrophoresis in Sanger, capillary electrophoresis. Basically, it's DNA sequencer, and then that output the DNA sequences, right? So these days, after PCR, most of us, what we do is we send the sample for sequencing to the company, right? Outsourcing. And the company, they will take a week or two, and then they email you back the AB1 file. These files are sequence files, and then it's up to you to how you are going to analyze it. Right. So in the DNA taxonomy, first step is to get the sequences. That is basically the gene or intergenic locus. It could be one locus or multiple locus. So more than one locus is nowadays preferred. for more precision in your statements of your papers. And then, well, to get the sequence, well, if you are not doing any wet lab work, if you're, for example, you're a college student, right? You just want to know how to do all this in Mega, so you can do it. You can form a very interesting hypothesis. For example, the evolution of certain trait in uh, plants. For example, leg hemoglobin. Is the leg hemoglobin evolved through horizontal gene transfer mediated by transposon? That is one big question, right? So to know that, you don't even have to go any sampling, right? So all you need to do is download from the NCBI, right? NCBI nucleotide, and analyze in your laptop using Mega, and then interpret the results and write a paper. You can publish it. You don't really need to go do any lab work. So it is really economical for resource delimited conditions, like in many non-affluent colleges of India. That is why this workshop makes sense in the colleges like yours. I know the government institutions are underfunded, right? And yeah, so in silico works like that are much easier to do, and many people are publishing high quality papers solely based on the in silico work. That is absolutely ethical and absolutely fine too, right? So the step one is to get the sequence, either your own sequence or you can get from the NCBI. So, well, inside Mega, there is already a built-in, the browser where you can download directly. You don't need to open Edge or Chrome and download FASTA file text and then exporting, importing into Mega. No, nothing is needed, right? It's much more simpler. Second step is to align the sequences. Align is basically to see the conserved sites. So certain sites are conserved inside the gene or locus. Certain sites are conserved across the species that we look. So we need to align such a way that these conserved sites are in column, right? So that is called alignment. So alignment is performed by introducing the gaps if necessary. So basically, gap means mutations, isn't it? In insertions or deletions are called gap, isn't it? So yeah, you need to introduce the gaps. So aligning sequences is done through something called multiple sequence alignment. So there are several algorithms available like Crystal W, Muscle, and all. You can do that in the Mega. Muscle is super fast and pretty good too. You can just use any of these. Next up, once you have aligned alignment ready, you can trim the ends, primer binding site and the tail region, which is not really good with the sequencing, of homopolymer dimers you might get in the end. So like TTTTTT, it kind of slur, right? So you can simply delete all those region at the end of your sequence. So once you have an alignment ready, next step is to find the best fitting model. So in any phylogenetic inference, there is something called model and method. What is the difference? Model is an equation. It's a mathematical equation. And usually these equations are really complicated, third order or quadratic equations. So a high level of mathematics is needed in order for you to understand what these models are. So you can skip it, no problem. Well, understand there are so many equations, around 25, 30 different equations, and each equations have a lot of parameter can be further changed, further modified. Okay, so there are models, but out of these model, which model is best for our data? What is our data? Multiple sequence alignment, right? With that data, among, let us say 50 equations, which equation can best explain our data? So that choice has to be decided based on a test, a proper statistical test. So in the Mega, you can do something called ML model test, that is maximum likelihood model test. So basically this test is an implementation of hierarchical ratio test, HLRT, likelihood ratio test. Hierarchically, you are performing the likelihood ratio. So it's a mathematical operation. So at the end of it, it will give you a table based on the Bayesian information criterion or Akaike information criterion, and all the 50 equations will be arranged in ascending order, right, or rather descending order of the best fit. So the top model is the best model, you see. So you need to find that step is very crucial. So once you know which model to use, then coming the next step can be a distance matrix step. You can construct a distance matrix. So distance matrix, a good example would be just check out any geographical map, like for example school atlas or map of Andhra Pradesh, right? Different cities like Kakinada to Visakhapatnam or Kakinada to Nellore, right? How much the difference? So Nellore to Kakinada is exactly same as Kakinada to Nellore, isn't it? So that is why this distance matrix is mirror like. So you don't really need the other side, you just need to put in one side. And that is why the distance matrix usually have a triangular shape, right? I hope you know what I'm saying about. So otherwise, just why not? You can just check out any map on the left or right corner. You can see the distance matrix in a triangular form. So same way we can construct a distance matrix of our species, that is our DNA sequences. Like for example, human being to chimpanzee, how much the distance? Human being to elephant, what is the distance? You can construct it. Remember, distance matrix approach is an extension of phonetics, the term we covered in our first talk, phonetics and cladistics. So phonetics method depends on the distance matrix. So this is our input. From MSA, multiple sequence alignment, a distance matrix is constructed, and from distance matrix, a tree is constructed by UPGMA or neighbor joining method, right? Now, fifth step is to do a phylogenetic analysis. So basically, there is a tree construction using any kind of method, right? You can use all these things in a software like POP or Genius, Phylip or Mega. Many softwares are available. Mega is a very good software for learners because it's completely free and it is pretty powerful. And Sudhir is a developer of Mega, is actually an Indian, you know. Yeah, so that is the fifth step, to construct the tree using any method of phylogenetic inference. So there are models, there are methods. Models are equations, while method is the approach. Is it phonetic or cladistic? If it is phonetic, is it neighbor joining or UPGMA? If it is cladistics, it could be maximum parsimony or maximum likelihood or Bayesian inference. So these three are very popular for cladistic method. And remember, I told you cladistic methods are much more accurate than the neighbor joining and UPGMA, these phonetic methods. Okay, and these days nobody does phylogenetic inference with neighbor joining and all. It became really obsolete. Yeah. So, well, to tell you an overview, maximum parsimony is, you know what parsimony means? Parsimony in the English word, it means miserly, frugal, don't want to spend money, right? Conjuice, isn't it? So maximum parsimony approach, the philosophy is that the best tree is the tree with minimum number of steps. With a set of sequences, the multiple sequence alignment of, let us say, 5 species, you're constructing a tree. So you can have, let us say, thousands of trees with that 5, right? Depends on how they are actually clustered. So with the 1,000 trees, which tree is the best tree? So according to maximum parsimony, the best tree is the tree which need less assumptions, that is less number of steps, right? So that itself is an extension of a philosophical concept called Occam's razor, like shaving razor. If one blade razor can give you a very close shave, why you need to go for 2 blade razor? How about 3, 4, 5, 6? Even 6 blade razors are available in Amazon. Even 12 blade, 16 blade razor, can you believe, in some of the foreign Amazon like Amazon Germany. There is no point on going for unnecessary complication if simple solutions are optimal, isn't it? That is the philosophical rule of thumb called Occam's razor. That concept is used in phylogeny too. Maximum parsimony approach is a classic example on it. And Bayesian approach is a completely different approach. Bayesian approach in statistics, it depends on the posterior probability or prior probabilities. So, you know that the probabilities are not fixed, like you're tossing a coin, right? So it can land either on head or tail. Probability on landing on head is 50. Tail is also 50. So it doesn't matter how many times you're tossing the coin, right? 100 times you're getting head. Every single thing is head. Now 101 time, what is the probability of head or tail? Same, 50-50. It doesn't depend on the previous. But in reality, yeah, it depends, isn't it? Like if it rained in Visakhapatnam today, what is the chance of rain tomorrow? Is it 50-50? No, there is a high chance, isn't it? So yeah, so it depends on posterior assumptions. So we keep on changing the probability when we come across the new information. That is the Bayesian approach. We keep on changing. That is how the human beings we think. You know, for example, if I give you a middle-aged lady's picture, an Indian lady, a Telugu woman, and I say that okay, she is a graduate from Oxford University in sociology, and her IQ level is very high, and yeah, very smart lady, and she is contesting in elections in Visakhapatnam, right? Will you vote for her? That is the first question. Well, you might say, well, yeah, I will vote for her. Looks to be fine. She seems to be capable, you know. Then I say, okay, let me tell you a little bit more about this lady. She is very corrupt. Will you vote for her? Suddenly you might have changed your decision to vote, right? You will say, no, I will not vote. So you keep on updating your probability whenever new information is provided. That is called Bayesian approach. Okay. So yeah, there are several approaches on phylogenetic reconstruction. You can construct based on the approach. And these days, the best approach seems to be maximum likelihood and Bayesian inference with an edge going towards the BI, Bayesian inference. Many top articles are based on Bayesian inference. Okay. So this is a worked out example of the DNA taxonomy of a few plant species using RBCL gene in the mega. Let us do it. Okay. So I'm just yeah, I'm just stop sharing this particular. I can just hide it. I think I can just exit. You can still see my screen. I hope my screen is still visible to all of you.
Yes, sir.
Okay.
Yes, sir.
So now let me open Mega. Oh no, it's not coming up here. Yeah. So I opened Mega. So I suggest all of you, if you have a laptop, if you're working with a laptop, you can open the Mega just like along with me. Okay. So it becomes a bit more clear. So I'm using a magnifier. Okay, so this is the Mega. I think the screen is visible to all of you. And yeah, so see, well, first step is that we need to get a few sequences. So how do you get a sequence? So you need to click on align. Okay, so click on the align and then query data banks, this option. Fine, I click on the query data banks. I'll just yeah. Now this is coming up, right? NIH, NCBI, nucleotide is coming up here. So this is how the NCBI. I'm sure every one of you have seen this. So don't have to change anything. You just need to search certain plant and one locus. Remember, I told you we are going to look at RBCL, right? So okay, now let us search mango. You don't even have to put Mangifera indica, no, just put mango and put RBCL. This is a gene. Is that clear? Mango RBCL, and I'm searching out. So now you can see that well, the first one is a reference sequence. Well, forget it. Items 1 to 20 of 104. That means there are 104 accessions of mango RBCL. Now you can see the first one, you can see 551 base pair. Second one is also 551. If you scroll down, you can see something longer. Let's go with a bit longer, like 946 base pair. Longer means more information, more sites. Okay. So what I do is that I click on GenBank, not FASTA, not graphics, not PopSet. I'm clicking on GenBank. You can also click onto it, GenBank. So if I click onto it, this is our Mangifera indica, RBCL, GenBank. So this is our sequence. You can see that it's around 946 length, right? So this is the first sequence. So now what is the step? Here on the top toolbar, you can see add to alignment with a plus, a red plus sign with add to alignment, right? Can you see it? Yeah, I
Yes, sir.
click onto it, add to alignment. Okay. So the two options come: import only, import full sequence. Well, I like full sequence, but this time we'll go with import only. No problem. I click OK. And then it will say first word, second word, and third word. So basically these words are on your alignment, how this one is being arranged. Okay. The strings of the sequence name. So first word, let it be binomen, that is Mangifera indica. Second word, let it be the locus, that is RBCL. And the third word, let it be our accession number, MK264366. Okay. So now the alignment explorer window has opened up. This is our main, this is a browser window. Multiple windows are there. Don't worry about it. Okay. This is an alignment window of the Mega. Okay. So now we have only one sequence inside the alignment window. Are you speaking? Somebody, please, please mute. Right. Okay. So now let's go for another plant, another tree. Let us say cashew. So cashew RBCL. So this way you can just populate it. Okay, any number of tree or any number of plants, you can just keep on searching. So first one is 754 base pair, seems to be fine. So let us click on the GenBank. Then I click add to alignment. Then I click OK. Then I need to say first word, second word. This you need to do every time. Okay. Second word is RBCL. Third word is our GenBank identifier. I click here. Okay. So now you see that we have now two sequences here. First one is Mangifera indica. Second one is Anacardium occidentale. That is our cashew. So let us go back here. We'll check another one. Let us say pistachio RBCL.
Excuse me, sir.
Yes.
How do you go back to get another sequence?
Or just go to the same browser. No, you don't have to close that browser window.
Okay.
Just go to the browser and search it. That's all.
Okay, sir. Thank you, sir. Sorry.
Yeah. So I searched in the same browser. I searched for pistachio RBCL. So scroll down. The first hit is Pistacia vera. That is a binomen of the pistachio RBCL. And I don't have to, as I told you, don't click here. You have to click directly on GenBank. And then I click add to alignment. I click OK. And then first word is Pistachia vera. Second word is RBCL. And the third word is GenBank. I click here OK. Yeah, well, now we have got 3 species in it. right? And now let us also add one more species as an outgroup. Remember what is outgroup? So whichever family these three are in, we need a plant which is not part of this family. Any guesses what is the family of these three species? It's Anacardiaceae, right? So that is a plant family of pistachio, cashew, and mango. So let us choose, let us say sunflower. Sunflower is not part, it's a part of Asteraceae, isn't it? So I'm searching sunflower or BCL in the main window. Okay, so the first hit is very small, 295. I don't want it. Second one is 553. If you scroll down, yeah, you're getting bigger. See, 801. And I click here, GenBank. I click add to alignment. Okay. First word the same way, Helianthus anus, then RBCL, then accession number. I click here, okay. Yeah, fine. Now, what is it? Now we have four species here. This is our outgroup, Helianthus. Sunflower is outgroup because it is not part of the in-group, the Anacardiaceae, right? And now the next step is aligning the sequence. So construction of the multiple sequence alignment. How do I do that? I'm just magnifying a little bit. So here you can see alignment option. One is W and one is kind of a muscle, biceps, right? So you can choose any of these, either cluster W algorithm or muscle algorithm you can use, right? Let us use muscle algorithm. I just click here muscle, and then there are two options coming: align DNA or align codon. Do you want to align by codon? Codon is basically, coding, the triplet codon. So let us simply go with align DNA. That is a very simple approach, right? Nothing selected for the alignment. Do you want to select all? Yeah, I want to select all. And you don't have to change anything, just accept the default delimiter. I click here, okay. And now it is aligned properly, okay. So I can just reduce that thing. Yeah, it is now aligned. I can just scroll up. It's kind of too big. Yeah, this sequence is pretty big. The final sequence, no? While these sequences are very less. So it makes sense to carefully look the alignment, how good the alignment looks like. Okay. Anyway, we can just try with one more cluster W alignment. I click here, okay. Again, it's the same alignment. I'm not satisfied with this alignment. Anyway, it should be like should look like a block. Okay, only this area has some block, but the rest of it is just nothing, right? So it doesn't look like a very good alignment to me. Maybe some issues with these sequences. I don't know why. You need to work on it, right? Anyway, now we have few things. So now let us actually work on the same thing and let us actually see that how do you do that. So once the alignment is completed, once you construct this alignment, next step is to do the phylogenetic analysis, right? So for that, you need to click on data and click phylogenetic analysis. That is very, very important. I click here data and phylogenetic analysis. Now it's asking, is it protein coding nucleotide sequence data or not? So well, remember this one is RBCL, right? RBCL is a protein coding nucleotide sequence. So I have to click yes. Had it been an intron like an ITS, I would say no, it is not a protein coding intron. It's our main window of the mega. So in the main window, first step on doing this phylogenetic construction, remember I told you the the first step is to calculate the model. You need to find which is the best equation out of around 25 or 30 equations. So I click here model, find best DNA or protein model by ML method. Do you want to use currently active? You can just click on remember to use and I click yes. You don't need to change anything. I click here OK. Then the progress will happen. And then a table will be presented. You look at this table. So as per this table, T92 is the best equation. T92 is Tamura 92. So basically, a Japanese evolutionary biologist called Tamura published a paper in 1992, in which he described an equation with 7 parameters. So this equation is the best equation with its BIC value, Bayesian information criterion, Akaike information criterion, and LNL, log likelihood of this equation. All those data is here. Well, you just need to choose this T92 is the best option. As you can see, some equations have plus G plus I. All these are modifications. Plus G means inside the locus, some sites can have variable evolutionary rate. Plus I, that is G means gamma, the shape parameter. Plus I means invariable site assumptions. Certain sites in a gene are fixed. It will never change. So if you have those kind of assumptions, you need to choose this. In any case, our any equation, well, T92 can be further modified by plus G or plus I or plus G plus I. All this, see, plus G plus I is a variant of T92 equation. So in our case, is T92 is the best fitting model. That is the first step, right? Now the next step is to do the distance. The fourth option. If I magnify, you can see that the fourth option. The third option is model. Fourth option is distance. So in the fourth option, I click compute pairwise distances, the triangular distance, which I told you something like the map, isn't it? So I click here OK. So now you can see there is a matrix came up already. This is the distance matrix. I can just, yeah. So this is the distance matrix. As you can see that these are the distances. So, this is the mango, right? Number 1 is mango. So what does this mean? 0.1. This means this is the distance between, the helianthus and mango. So now anacardium versus mango, what is the distance? This is the distance, 0.98. Pairwise distances, right? And pistachia and mango, what is the distance? 0.98 again. While helianthus and mango, it is 0.1, right? So now what is that? This number 3, the third one here, 0.89 means helianthus, that is our sunflower, with the number 3. Number 3 is pistachio, right? That is the distance between these two. That is what it means. Okay. So, well, if in case you want to export that into the Excel, you can just just one click here, Excel export, and it will be displayed into the Excel directly. Okay. Yeah. So now you can see the distance in the Excel format. But in case you want to play with the Excel, well, that is how to do a pairwise distances. Is that clear to you? Okay.
Yes, sir.
Now let us do a phylogenetic tree construction. So I can just zoom in. So this option, like the tree, you can see a phylogenetic tree. Click on to it and then construct a tree to whichever method that you want to use. For example, neighbor joining, maximum likelihood, minimum evolution, UPGMA, all those things, right? So unfortunately, Bayesian inference is not supported in Mega. So for which you need to use PULP or Genius a software which I suggest is Genius Prime, which is a very, very nice software. It has a lot of other tool. If you're working with the DNA sequences, you can even do annotations with it, and also uploading the sequences to GenBank. If you're working with lots of barcode, that is the best software, though it is costly. Okay, so you can do any of this. Let us go with the first option, maximum likelihood, which is a very nice approach. Okay, so here the only thing you need to worry is this one, model or method. You need to click onto it and choose the right model. So which model you want to choose? Jukes Cantor, Kimura 2, Tamura 3, Hasegawa Kishino Yano model, Tamura Nei model, or generalized time reversible model. So Tamura Nei model is basically Tamura 92 model. So this is a model which I want to use, right? Tamura Nei, because we did a model test, and in that model test, it is it's clearly we got this result that Tamura Nei model or Tamura 92 model is the best fitting model, and that is why we are choosing Tamura Nei model for this. I hope it's clear. Now, just zoom out. I click here, OK. Oh, yeah, now you can see that this is a tree which came up. And as you can see that this tree is not accurate at all, right? Because Helianthus is an outgroup, but this outgroup is clustering with Mangifera indica, while Anacardium and Pistachia vera is grouping. So ideally, you should get mango, Anacardium, and Pistachia in one clade, and Helianthus as a different clade. So maybe you might have got this kind of group. So this is most likely because of the problem with one of the sequences. So you cannot rely every sequences in the GenBank. Some of these are wrong sequences altogether, right? So anyone can upload the sequences to the GenBank, right? Maybe this sequence is wrong, Mangifera indica, right? So, or it could be because of the forward and reverse, the read, right, in which frame you're reading, right? And maybe because of that, this kind of problems can happen. But anyway, this is how to generate the tree. And you can also this tree don't have any uh, uh, probability values, so well, you can construct that also. But now, well, there is a nifty feature inside the mega called caption. If you click on the caption, you will see that show in tree explorer, you can see that the caption automatically comes up, which tells you everything. How did you construct this tree? So the evolutionary history was inferred using maximum likelihood, using Tamura Nei model. The tree with highest log likelihood is shown, and heuristic search. Everything it's written in the this caption. Is a very nifty feature. And now, I didn't do any test of phylogeny in this tree. I can just discard it. I can just redo the tree once again with test of phylogeny. Let's go with neighbor joining method. So for test of phylogeny here, you can choose not none but bootstrap method and 500 bootstrap. Okay, so you're replicating the same tree 500 times, and 500 bootstrap might take a little bit time. Yeah, so now we have the test of phylogeny as well. Now you can see that there is this number displayed, and this, sorry, yeah, you can see 100. So Pistachia and Anacardium is forming a clade which is highly supported. The statistical support is very high. That is the meaning of this 100. Okay. Yeah. Is that clear to you, or I discard the result? I just close the rest of the files. Close the data. That is another option. You don't know how to save it. Yeah. Now this is like how you start the mega software. Okay. So once you start, this is a main window coming up here. And inside the mega, you can actually see it. Like for example, there are several of the example file inside the mega. How do you open the example file? So you can open a file or session, click onto it. Uh, well, you would need to navigate all the way to your this PC, maybe in data E, Windows C, the root folder, program files, and mega 11. So basically this is your installation directory, right? So this is where your mega is installed. Well, unfortunately, there is no example file in this version of mega which I'm using, but usually mega comes with example files. So this is icon only locales. Yeah, there is no example file in it. Very strange. Usually the mega comes with example file. Maybe I did some mistake while installing the software. Yeah, so you can navigate onto the installation folder and then you can click onto the
They
example
are
file.
lower section has examples.
Which one?
So in home menu, you can see in lower section there is example option in green.
In the main menu? Here the example. Oh, fine. Yeah, thank you for pointing out. So in the menu below, I can just I can just put it here, magnifier. So yeah, here there is example, right? So you can click onto it. This example. Yeah. So here you can see there are several example files coming up. Yeah, it's actually stored in a different folder, documents mega X. That is why it's not in the installation folder. It's inside the document. The file structure is very strange in Windows, isn't it? Sometimes it is in the root. Yeah, so now you can see that chloroplast, crab RNA, distance data, gene tree, HSP, mitochondrial cDNA, all those things are there. So let us open crab RNA. Okay, it's not a botany, but yeah, it's for the crab. I opened this one now. So now you can click on the alignment. You can see that there are different crab species, and this is our alignment file. You can see that this dot means same. So means this position, everything has got G. That is what is dot. So differences are highlighted in A or wherever the G is basically the mutation happens only for these two. So you can just scroll through it and see that how that data is organized. Right? It's very well, sequence alignment. Well, you can just minimize it. Then you can perform a phylogenetic tree. So you can construct. You can just check the model first. So find the best fitting model. I click here. Okay. Yeah. So according to this model test, HKY plus G is the best fitting model. Hasegawa Kishino-Yano with gamma substitution is the best equation. So now while making the tree, the phylogeny construction, now let's do with the neighbor joining. So inside the model, you can choose that particular model if it is available. So unfortunately, HKY is not available here. If such is the case, then you can go with the second best, Tamura 92, basically Tamura name model. Now let us go with Tamura name model. I click here, okay. Right now I can see that well, the trees with the bootstrap proportions, and the outgroup is Artemia, a crab species, you see. So yeah, this is how to construct the tree and how to perform the model test inside the mega, and how to interpret the tree. Yeah, so that's it. I hope things are clear to you. I don't need this result, I just discard it. Well, before going, I can just show you this also, just a small thing. Yeah, these are just name, right? The four species which we did, I copied everything. So I will just show you how to do this time tree. So the time calibrated tree which I told you earlier, just need to click on to this time tree on the right side of the a browser, main window, and find the divergence time between two taxons. Or if you put one taxon, you will be getting the evolutionary timeline. So that is another option. Or build a time tree. You need to specify, different species here, search and add, so that is an option. Another option is load a list of species. Let us choose that file and upload that file. So basically it is on my document. Okay, I have to go here. This is the document. It's basically a simple text file with whatever species that we are working on. Okay, I click here, upload. So now you can see this is a time tree. It's so simple to make this time tree. So the time tree has got an x-axis here, which is time. You can see this axis has got millions of years before present. 0 means present day, right? So for example, Pistachia, Anacardium and Mangifera, basically all these are our Anacardiaceae, evolved around this time. So this time is basically approximately 55 million years back, right? And as you can see, Anacardium and Mangifera is more related than with the Pistachio, but all these three are forming a clade with sunflower. So sunflower and this entire Anacardiaceae diverged very early on, around 125 million years back in Cretaceous. So these are the timeline, right? So yeah, this separation happened in Paleogene. This is Cretaceous. All these are part of the Phanerozoic eon. So these are eon, era, period, epochs, and age. You see? So yeah, that's how to make this time tree. I hope it's clear. Okay, so I'm open for any questions that you have.
Good afternoon, sir.
Yes.
Sir, you've mentioned that when we were taking the T92, sequencing model, how did you know which one was the best equation to take?
Yeah.
What are we considering?
So basically, you do a model test, right? I've shown you how to do a model test.
Yes, sir.
Yeah, which is basically based on hierarchical likelihood ratio test. It's a statistical approach. Okay, software do this test, and then based on that, it displays a table. So the table you simply picked up the top one. That's all. That is the best fitting model. Yeah.
Okay, sir. Sir, and I have a small question from the theory part as well. So, is the barcode, the DNA barcode, a sort of identity for a gene, or is it just used for authenticity and identification of species and all?
Yeah, well, identity basically, to identify a pre-existing relying on the barcode. And barcode-based identification is so much more powerful than simply looking at the morphology. The reason is that morphology can vary, depends on season, depends on ecological constraint, for example, grazer, and depends on the climate change, depends upon so many factors, isn't it? Even if the shore is exposed or is the shore is sheltered can influence the seaweed morphology, right? So relying on morphology alone is a very inferior way. And also characters are very limited in the case of like flower, you see. So floral formula, right? So maybe maximum 30 or 40 things you can, these are different parameters. But if you're doing a sequence, so one sequence will have 800, 900 characters, right? So all these are parameters. So it is much more informative if you do a DNA sequencing for species delimitation rather than simply relying on the morphology. And that is why many of these AI-based plant identifying apps, mobile apps like PlantNet, that is one of my favorite apps. Just you need to snap an image, especially for flowers. The angiosperm, just take a picture of the flower and upload into the PlantNet using the app. Basically, PlantNet is from American Botanical Society. It will tell you exactly which is that species, right? So that is based on AI-based, regression. So it has a model which is trained on example data which is already identified, right? So those data will from sunflower, they have a data of sunflower flowers. So now your flower, based on statistical regression, if that is matching the database, then they will say yes, it's a sunflower. So if you compare this kind of AI-based apps with barcoding is still so much superior, because this AI-based apps capture morphological data, right? A photograph is just morphology is being captured. So the parameters are very limited.
Omega can be used for any other, organisms, right? Other than just plants.
In any case, it's working only on morphology. So, yeah, Mega is just a common evolutionary, the phylogenetic software. So you can use it for bacteria, virus, whatnot, any organism on planet
Okay, sir.
Earth.
Sir, and can you put an unrelated root for a tree? Is that possible?
Come again?
Can you put an unrelated root for a tree, the phylogenetic relationship one?
Root is, it is not like randomly you're introducing root, right? So there are three methods of rooting which I explained. The first method is the most commonly used method is out-group rooting, right? So you are deciding a group which is not part of the in-group, right? Like for example, that sunflower, I purposefully added to root my Anacardiaceae in-group, right? So that is the most popular method of rooting. That is an unrelated, you see, unrelated to the in-group, like what you said, unrelated. So that is a method of rooting a phylogenetic tree. Now the question is that how unrelated?
Yes,
Right?
sir. That
For
is
example, Anacardiaceae, if you're working at that family, you have around, let us say, 15 species you're in group, and you have one out group, and somehow you decide, okay, let Cara be an out group. The terrestrial algae Cara.
yes, sir.
Is it a good out group? No, it is not. If the out group is too distant, it will have impact on the branching order of the tree. Right?
Yes.
So it should not be too distant, it should not be too similar. Both are problematic.
Yes, sir.
So you need maybe a good option is like Asteraceae, which is not really related with the Anacardiaceae. So that kind of decision has to be taken. Not too dissimilar, not too similar. Yeah.
Yes, sir. Just far enough from the family tree
Yeah.
is good.
Yes.
Okay, sir. Thank you,
And
sir.
it
Thank
has
you, sir.
more than one out group too. No problem.
Yes, sir. Thank you, sir. Thank you so much.
Sir, this is Neeraja Paspleti.
Okay.
I would like to know about the research work you have done so far using this DNA barcoding and especially in the DNA taxon.
Well, the work which my group has been doing is mostly concerned with the plants, the seaweeds, seaweeds of India. And so far, we discovered 7 new species using the DNA taxonomy, and all these species discoveries are multi-local. It's not just based on only one locus, but more than one locus. Yeah. So yeah, and apart from the 6 species of seaweeds from India, we also discovered one species of moss from Antarctic. So yeah, that those 7 are the species discoveries of using the DNA taxonomy, which I did. And then there are several new records, like for example, Sargassum zangii is invasive Sargassum species, earlier record only in China. Now we found it is there in Tamil Nadu coastline too. So that kind of new records, multiple papers we published. We also did work on blood rain phenomena in South India. In Kerala, we had a rain which is red color. So yeah, no one knows why it is red and why so. So we did the DNA taxonomy, DNA barcoding basically, and then the barcode turns out to be that of the algae. It's basically a green algae. It's very strange, right? Though the color is red, but the algae is green. Trentipolia annulata is a green algae, chlorophysium green algae. So the reason why it's red is it's basically spores of the algae, and spores are spread at very large area by means of rain. It's a spore dispersal mechanism, and spores have beta carotenoid. That is why it is red color. Yeah.
Excellent,
There
sir.
are multiple
Thank you
papers.
very much, sir. Excellent. Will you encourage any collaborations?
Yeah, well, why not? Any work that any one of you is interested from this Andhra, especially with the coastal region. So because my work is mostly based on marine botany, and yeah, the algae, especially the marine algae, really need a lot of attention. And
Yes, sir.
unfortunately, people are not really, it remains inconspicuous for most of us.
Because many people are talking about the biostimulants from the seaweeds.
Yeah.
So identification of the marine algae is very important nowadays.
Yeah.
So we are interested to work with you, sir, if you encourage us.
Yeah. Marine algae is really important, not just as a bioprospectives, but also like every breath we take. How many of you are aware of it? If you take 3 breaths, 2 comes from ocean produced by the algae. So in atmosphere, the oxygen around 68% is actually produced by picoplankton, cyanobacteria, marine cyanobacteria. But unfortunately, we don't really study about them, right? We are more interested in land plants, agriculture, and food for human being, right? Yeah. But without marine algae, none of us can live. Yeah.
Kindly give us your mail ID and phone numbers to be contacted.
Yes, sure.
Thank you so much.
Hi, sir. Good afternoon, sir.
Good afternoon.
Sir, I am from Karnataka, Punjab University, research scholar.
Okay.
Sir, I have one question. Sir, how can we find out the marker fidelity during how can we find out the marker fidelity of marker?
Well, fidelity of the marker. Well, you can actually do a lot of marker means basically the primers, right, for the amplification of the locus. So, well, there are the best option would be primer blast. If you're interested in to design a new primer, which is specific, so which is highly fidel, right? The fidelity is very high for the group in concern. Right, it doesn't amplify any other group, especially if you're worried about cross amplification and the problems like that. For example, Some of my PhD students are working on marine bacteria, right?
Okay.
And while culturing the marine bacteria, of course, there is a chance of cross contamination with the human pathogen. So we don't need that, right? We need a certain primer that can only amplify the marine bacteria, right? So if you want that kind of primer, yeah, primer blast is the best option. Just go to the primer blast and then choose the specificity criterion at which taxonomic level it has to be specific. Then
Okay, sir.
the software, basically, that it's just a website. It can tell you the forward and reverse primer based on its annealing temperature. Yeah.
Okay. Sir, I have another question.
Yeah.
So during posterior probability analysis, for how much n generation you have to give for getting a good tree?
How many iterations?
N generation. Yes.
Yeah, maybe around 100 could be like a minimum. So
100, sir.
yeah, 100 could be minimum. Well, it can go up to 500. Well, the point is that if you keep on increasing the replications, then it will become really long, right? Unless you have a very powerful computer, especially if you're working with lots of taxa, it is computationally expensive. That's why, yeah. Iterations can be, n can be 100 as the bare minimum, so then it should be fine. Yeah.
It's last question, sir. Sir, on which basis we have to select the outgroup species?
Well, as I told you, it should not be too distant. It should not be too near. So you just need a balance, So yeah, very near. Like if you see the tree of life, there is a barcode, the ITOL is available, right? Interactive tree of life. So you can just zoom into your family in concern if you're working with certain plant family. So in that family, just next family, maybe a sister family, don't choose it, which is very near to your family. So
Okay,
choose a family little bit away, but not too away also, not like a pine way too distant, right?
Okay, sir. Thank you. So can you suggest some softwares regarding this maximum Boltzmann analysis and posterior probability analysis?
For ML, the gold standard is PAUP, PAUP.
Okay.
Yeah, and yeah, so all these things you can do that using Genius, which I told you. Genius supports maximum parsimony and maximum likelihood and Bayesian probability, everything it does.
Okay.
Yeah.
Thank you, sir. Thank you so much.
So your university is called Kuvempu.
Yes, sir. Kuvempu
Very
University.
famous poet, right? The Kannadiga poet, right? Yeah.
Yes, Kuvempu.
Hmm.
Thank you, sir.
I think there are some questions. I didn't see that. What is the question on the chat box? Sir, if there is no any data of gene bank of species we have, how can we take step towards for that, sir? Well, if you don't have any species in GenBank for the what you're working on, so the only option you have is that you can work with the holotype of that species, right? Holotype is the herbarium sample, the basically herbarium sheet, the voucher, which is deposited while when you read the paper, the description of that species, right? Whoever discovered that species, there will be a formal description. So in that description, invariably the author might have said that holotype, designated holotype voucher accession number is this, deposited in this herbarium. Let us say Uppsala herbarium in Sweden. So the holotype of mango is in Uppsala herbarium. So if you really want to work on that mango, so let us say mango don't have any sequences in the GenBank, but you really want to work on the mango sequence. Only option you have is to write to that Uppsala University herbarium to loan you a little bit of fragment from that voucher. You need to explain it with all the detail, and also you need to enclose your capability by putting your papers, published papers, So then the herbarium can able to loan you a fragment of the voucher. Yeah.
Hello, sir.
Yeah.
Sir, I have to choose one particular genus for my phylogeny and molecular taxonomy work.
Okay.
In that genus, I have only 4 species are there, sir. Can I construct the tree by using that 4 species?
Yeah, there's no harm
Only
in it.
4
Only
species.
4 you can construct a tree, no problem.
Okay. Is there any chance to take some associated species from that genus?
Yeah, GenBank you can take it. Yeah, that is usually that makes more sense, right? Your in group, your the plants that you work with, how they are clustering within non-species from elsewhere, right? So you can download the sequences, which is so basically these 4 species of yours, each one you can blast and download the top 3-4 hits, and then all these together you make a multiple sequence alignment, and then you can do the phylogenetic analysis. So that is the best option.
Okay. Okay, sir. Thank you, sir.
Any further questions?
So Prashant asked, where can we take sample holotype? What is the procedure we need to follow? I explained that procedure now. So you need to write to the herbarium for working with the holotype. It is tricky scenario. If you have no species, well, if that species is non-species but no barcodes are available, then this is the only option to confirm that you have the sequence there.
Okay. Thank you, sir. Your hands-on training session was incredibly valuable. Everyone gained a deeper understanding of Mega software and its applications. Thank you for your patience and expertise. We are truly grateful for your time and effort in conducting these sessions. Thank you very much, sir.
So thank you so much.
Let's take a quick 30 minutes break for lunch.
We will resume it.
We will resume at 2:15 p.m. Thank you.
A new meeting.
A new meeting link will be shared closer to the start time. Enjoy your lunch. Thank you.
ఇక్కడ ఫొటోస్ అన్ని ఎందులో క్యాంప్స్ క్యాంప్స్ వెళ్తాం తీసుకెళ్తున్నాం ఎక్కడ పడితే అక్కడికి వెళ్తున్నాం మేడం కరెక్ట్ డిస్కనెక్ట్ చేయమంటారా అది అయిపోయింది మేడం అప్లోడ్ చేస్తాం The